calorie is the amount of energy needed to raise the temperature of 1 gram of water to 1 degree Celsius. Glycolysis is the cycle of releasing only a small amount of energy. Cellular respiration is the process that releases energy by breaking down glucose and other food molecules in the presence of oxygen. The equation for this is oxygen, sugars becoming carbon dioxide, water, and energy.
Glycolysis is the process in which one molecule of glucose is broken in half, producing 2 molecules of pyruvic acid, a 3-carbon compound. ATP production is the main source of the energy for glycolysis. The NADH production. One of the reactions, glycolysis removes 4 high-energy electrons and passes them to an electron carrier called NAD+, a nicotinamide adenine dinucleotide.
There are 2 types of fermentation. Alcoholic fermentation gets pyruvic acid and NADH into alcohol and carbon dioxide and NAD+. Lactic acid fermentation gets pyruvic acid and NADH into lactic acid and NAD+.
The Krebs cycle was named after Hans Krebs, a British biochemist, in 1937. During the Krebs cycle, the pyruvic acid broken down into carbon dioxide in a series of energy extracting reactions.
The Krebs cycle begins when pyruvic acid produced by glycolysis enters the mitochondria. 1 carbon atom from pyruvic acid becomes part of the molecule of carbon dioxide, which is eventually released in the air. As the cycle continues, citric acid is broken down into 4 carbon molecules. Then carbon dioxide is released and an electron is transferred to energy carriers.
The electron transport chain uses the high-energy electrons from the Krebs cycle to convert ADP2 into ATP. The high-energy electron from NADH and FADH2 are passed along the electron transport chain. The electron transport chain is composed of a series of carrier proteins located in the inner membrane of mitochondria.
Every time 2 high-energy electrons transport down the electron transport chain, their energy is used to transport hydrogen ions. The ions then rush back across the membrane, producing enough force to spin the ATP synthase and generate enormous amounts of ATP.